Hi everybody, and welcome to our module on ketone bodies. Ketone bodies are an alternative fuel source for some cells. So just like some cells can use glucose and fatty acids for energy, some of them can also use ketone bodies. We'll talk about their structure in a minute. In times of fasting or starvation, as you know, many fatty acids are sent to the liver, and the liver is where ketone bodies are synthesized. So during these times of fasting or starvation, the fatty acids will lead to high levels of acetyl-CoA in the liver via the process of beta oxidation. The high levels of acetyl-CoA can exceed the capacity of the TCA cycle, and as a result, levels of acetyl-CoA can climb. When this happens, acetyl-CoA will be shunted towards ketone body synthesis. And I've shown you a picture of that at the bottom of the screen. The fatty acids increase the amount of acetyl-CoA inside of liver cells. This exceeds the capacity of the TCA cycle, and thus acetyl-CoA molecules are shunted towards ketone body synthesis. On this screen, I've showed the pathway for synthesis of ketone bodies, and it begins by combining two molecules of acetyl-CoA. And that should make sense to you because I just told you that these are synthesized during times when there are high levels of acetyl-CoA present in liver cells. The first step involves formation of a molecule of acetyl-CoA, which is basically an acetyl-CoA molecule with an extra carbon group attached. The next step involves formation of HMG-CoA. HMG-CoA is hydroxymethylgluteryl-CoA. It's this large molecule I've shown on the screen here. It is also a very important intermediate in the cholesterol synthesis pathway. From HMG-CoA, a molecule of acetoacetate is synthesized. Acetoacetate is the first ketone body that is synthesized. It's one of the ketone bodies that can be used as fuel. This is its structure right here. And then acetoacetate can be broken down into 3-hydroxybutyrate, which is this structure on the right side of the screen, and also acetone. Now, acetone is interesting because it cannot be used as fuel, so it is not a ketone body that's used to derive energy by cells. However, it is produced in the ketone body synthesis pathway. Another interesting thing about acetone is that it is not excreted in the kidneys, it is excreted in the lungs. You exhale it, and that is why patients who are synthesizing ketone bodies have fruity smell on their breath. Ketolysis is the process by which 3-hydroxybutyrate and acetoacetate are converted into ATP. Remember, I said these are the two ketone bodies that can be used to generate energy by cells. The liver releases these ketones into the plasma, and there is a constant low level of synthesis going on all the time inside of your body. But the synthesis increases in fasting when fatty acid levels are high and the level of acetyl-CoA rises inside of liver cells. The ketone bodies that are released by the liver are used largely by the muscle and heart, and this spares the glucose for use by the brain in times of fasting. Now, the brain can also use some ketone bodies during times of fasting, but importantly, the liver cannot use ketone bodies. This means the liver is going to synthesize these structures, but it cannot metabolize them, so it has to release them into the bloodstream so that they can be used by tissues like the muscle and heart, and sometimes by the brain. This is the process of ketolysis. It's essentially a reverse of the process of ketone body synthesis. It begins with 3-hydroxybutyrate. This is converted into acetoacetate, and then eventually acetoacetate degrades into two molecules of acetyl-CoA, which can then go and enter the TCA cycle in tissues like the muscle, heart, and the brain. The big picture of ketone bodies that you should understand is that the brain cannot use fatty acids for energy in times of fasting or starvation. The reasons for this are poorly understood. It used to be believed that fatty acids could not cross the blood-brain barrier. There's now evidence that they can. It may be that brain cells that use fatty acids were subjected to oxidative damage, and this created an evolutionary pressure so that the brain doesn't use fatty acids in times of starvation. I've shown a nice review article on the bottom of the screen here if you want to learn more about the reasons that are hypothesized to explain why the brain doesn't use fatty acids. But for whatever reason, the brain cannot use fatty acids during times of fasting or starvation. So ketone bodies allow use of fatty acid energy by the brain. In other words, fatty acids are converted to ketone bodies in the liver, and then those ketone bodies go to the brain, which can use them as acetyl-CoA. This allows the fatty acid energy to be sent to the brain in terms of acetyl-CoA. In addition, the ketone bodies are used by other tissues like the muscles and the heart, and this preserves glucose for the brain because once again, the brain can't use those fatty acids, which are very high sources of energy during times of fasting and starvation. Ketone bodies have a low pKa. This means they tend to release protons and act as acids at plasma pH. If you look at the structure of acetoacetate on the screen here, both of these single bonded oxygens can release a proton and thus lower the pH of plasma. In addition, 3-hydroxybutyrate has a COOH group here, which can also release a proton and lower the pH of plasma. This means when you have high levels of ketones in the plasma, you can develop an anion gap metabolic acidosis. Now, fasting and starvation aren't the only situations where you can see high levels of ketones in the plasma. A very important clinical condition where there can be high levels of ketone body synthesis is diabetes. Diabetics have low insulin levels. This means they will have a high utilization of fatty acids. In addition, diabetics can deplete oxaloacetate from the TCA cycle and thus stall the TCA cycle. Let's look at that in a little more detail. On the screen here, I've shown the first step of the TCA cycle where acetyl-CoA combines with oxaloacetate to form citrate. In the setting of low insulin levels in diabetes, much of this oxaloacetate is being diverted to glucose production for gluconeogenesis. Thus, that decreases the pool of oxaloacetate and slows down the TCA cycle. In addition, fatty acid oxidation produces a lot of NADH. This tends to drive this step of oxaloacetate and malate in the reverse direction. This is a reversible step of the TCA cycle. So in the setting of high levels of NADH, it will be driven towards malate, and that will also lower the oxaloacetate level. So for both of these reasons, there will be relatively low amounts of oxaloacetate present inside of cells, and this will lower the rate of acetyl-CoA uptake by the TCA cycle, and thus the levels of acetyl-CoA will start to rise, and that increases the synthesis of ketones. I've summarized this for you here on the slide. The TCA cycle stalls, there are high levels of acetyl-CoA, and ketone production can occur. This is a big problem when diabetics get an infection and their insulin requirements rise, or if they skip dosages of their insulin. In both of those situations, they can have relatively low levels of insulin, and thus they can get high levels of ketones in their plasma, which can make them acidotic. This is a condition called diabetic ketoacidosis. Another clinical condition where you see high levels of ketones is in alcoholism. So the metabolism of ethanol involves the production of NADH, and thus there will be excess NADH inside of the cells of patients who are consuming large amounts of alcohol. Thus, the oxaloacetate will be shunted to malate, just like I described in the last slide. There'll be lots of NADH here. This means this oxaloacetate will be favored to go in the direction of malate by the enzyme malate dehydrogenase, which catalyzes this reversible reaction here. This will stall the TCA cycle, and thus there will be higher levels of acetyl-CoA present. This will favor ketone body production. Incidentally, the high levels of NADH can deplete oxaloacetate for gluconeogenesis, and that's one of the reasons that alcoholics can show up to emergency rooms hypoglycemic, in addition to having high levels of ketones in their plasma. You can measure the plasma levels of ketone bodies, but this is rarely done. Usually the way you will detect ketone bodies is in the urine. This is a picture of a urine dipstick, and you can dip it in a sample of urine, and it will change color to indicate the presence of ketones in the urine. Normally there are no ketones in the urine. Any that are produced are usually utilized by the body. So if you detect them in the urine, it means that something is wrong. You will see this very commonly when you're working in emergency rooms and other clinical settings where you test the urine of patients with different clinical conditions. You can see elevated urine ketones in patients who have poorly controlled diabetes. They have insufficient insulin, as we just described 2 slides ago, also in diabetic ketoacidosis. Patients who have not been eating enough for a long period of time will have high ketones. So people who come in with extensive nausea and vomiting may be very undernourished, and therefore they can have ketone bodies present in their urine. And then, as I said before, alcoholism can lead to the production of ketone bodies that can be detected in the urine. And that concludes our module on ketone bodies.